Hi, everyone, and welcome to today's program, Your Inner Neanderthal, a deep dive into the Neanderthal and Denisovan DNA in modern human genomes, featuring geneticist Laurits Skov, and this is part of our ongoing Hot Topics series. My name is Briana Pobiner, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray-haired woman wearing a red shirt in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us or you've attended before, we're so glad to have you here. Before we get started, I have a few housekeeping notes. This discussion offers closed captioning. You can turn the captions on or off via the CC button, which should be located at the bottom of the Zoom interface. We're in a webinar format. We can't see or hear you. So as you have questions, please go ahead and submit them to the Q&A box, which is at the top or bottom of your screen. It looks like two speech bubbles, so we can sort through as many questions as possible. The Q&A time really flies by. The Q&A box is also where we will share any relevant links during the program, so keep an eye out there. We'll start with an opening presentation by our speaker, Dr. Laurits Skov, and then I'll join him here to take your questions. During the presentation, I will also write answers to some of your questions, as will one of our behind-the-scenes staff, Dr. Ryan McCrae, at least any that we can answer. So now I'd like to go ahead and introduce our speaker. Dr. Laurits Skov is a scientist interested in Neanderthals, both how they lived and how their genes survive in us today in present humans. Laurits did his PhD in bioinformatics at Aarhus University in Denmark. And during his PhD, he developed a method for detecting Neanderthal and Denisovan ancestry in modern humans without using archaic reference genomes. And he used it to find archaic segments in Icelanders and Papuans. He then did a postdoctoral fellowship at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, where he further studied archaic introgression in ancient genomes, and he studied Neanderthal communities. He's currently a postdoctoral fellow at the University of California, Berkeley, where he's focusing on studying archaic introgression in South Asian populations and the evolution of human germline mutation rate. So now I'd like to welcome Dr. Skov to the screen, and we're looking forward to his presentation.
Thank you very much for that introduction. So we had some technical issues today, so I won't be in charge of my own slides, but hopefully this will work out just fine. So this is what we're going to talk about today. What I'm showing you here is a Neanderthal father and his daughter. And Neanderthals are not around anymore. They are extinct. But in some sense, they are still around and they are still surviving. And they're surviving through the DNA they passed on to us. Next slide. Yes. So the questions we're going to be talking about today, next slide, will be sort of which of these types of humans, so Neanderthals and Denisovans, did we modern humans meet with, and what happened in that exchange. So we got some of their DNA, some of their genes, and some of these pieces of DNA have a function and it has an impact in our lives today. And we're going to be discussing all of that. So we're living in quite extraordinary times, actually, in the sense that we are the only type of human alive today. If you go back 100,000 years, there were at least 7 different types of humans alive all over the world. And I sort of annotated them here on this map. So in the big gray circle, we have early modern humans, at this time primarily living in Africa. We have the Neanderthals, primarily living in Europe and West Eurasia in blue. We have the Denisovans in orange, primarily located in East Asia. And then we have a lot of other human groups from which we have no DNA. So it's hard to place them in our family tree. And the reason for that is it's very hard to find DNA when the climate gets hotter, DNA breaks down. Can you go to the next slide? Oh, wait, actually the one before, sorry. So that was the situation 100,000 years ago. But then 50,000 years ago, things start to change. And it changes in a sense that a group of humans from Africa leave Africa and go on to expand into the entire world from there. And and that gives rise to everyone with non-African ancestry. And as this happens, all other types of humans go extinct. So leaving only us today. But in the past 20 years, there's been sort of the field of ancient DNA has emerged. Like there's been a giant revolution in the techniques we have. So we actually been able to get DNA from the Neanderthals, from the Denisovans, from skeletons that are more than 50,000 years old. And next slide. And just to show you how amazing this is, so this is like a little picture of the bones that we got DNA from. So for instance, for a Neanderthal, it's just like a small bone of the toe. And for the Denisovan, the bone that is Denisovan DNA is a pinky bone. It's one of the smallest bones in your body, and it's the bone right on your little finger. So from just looking at the bone, it's hard to say there's another type of human, but sequencing DNA, you can find that out. So now we're going to be talking a lot about DNA. So just, I'm sure this will be familiar to most people, but just a quick summary, so we're all up to speed. So our DNA is organized in our cells into something called chromosomes, and most people will have 23 chromosomes. You will get one chromosome from your father, and you will get one chromosome from your mother. And these chromosomes, they are just long stretches of this double helix type structure. And it consists of 4 different types of bases, 4 chemically different types of molecules called bases. And these bases we just call A, C, G, and T for short. And you could sort of imagine this as if you imagine it as a book, this book would be more than 3 billion letters long. And you could say you get one book from your mom and one book from your dad. And next slide. So what happens with when our cells replicate, it's like they make a copy of the DNA, so that your cells are replicating all the time. But sometimes this replication is not perfect. It introduces errors. And every person alive today will have something like 75 new mutations passed on from their parents. Or can you actually go back a slide again? So there's all these mutations, and sometimes these mutations happen in the germ cells, so the egg cells or the sperm cells, and then they're passed on to the next generation. And this process happens through time, and this is what's making species and different types of people genetically distinct from each other. Next slide. So if you take 2 humans, for instance, like most of the time, 2 humans are going to be identical. Their DNA sequences are going to be identical. There's going to be something like 1 difference every 1,400 base pairs. But if you add a chimpanzee, for instance, you see that their DNA is something like 10 times more different than between 2 humans. So if you take Neanderthals, you would see that Neanderthals are something like twice as different as any 2 humans. So you can sort of make a family tree out of that. So chimpanzee would be more distantly related to humans, and Neanderthals would then be closer related to humans. And so we can actually make a family tree like from the DNA sequences. And what we found is that humans, and I'm showing you here on the left, there's different groups of humans living different places in the world. We all share a common ancestor with Neanderthals, something like 600,000 years ago. And then what we also learned from, one slide back, then what we also learned from studying the Denisovan genome is that these Neanderthals and Denisovans are closely related to each other. So you can sort of think of them as the Eastern and Western version of some archaic humans. And they have a common ancestor of 400,000 years ago. But this is not the complete picture. It's not these populations and then they split and they never interact again. When we sequenced the genomes of these Neanderthals and Denisovans, we actually found out that it's more complicated than this. So they contributed DNA to us. Next slide. So it turns out that everyone who has non-African ancestry has some Neanderthal too. And it's something like 2% of your genome that is inferred to be Neanderthal. And because it is in all non-Africans, we speculate that it might have happened somewhere in the Middle East, when you move out of Africa, somewhere soon after that, before people move into Europe and East Asia and Oceania and the Americas and so on. And if you go to the next slide, you see that there's also evidence that we also met with the Denisovans. So they also contributed DNA to us. For most people, can you go back again? For East Asians and Europeans, this is a very small amount, something like 0.1%. But for people in Oceania, so people from Papua New Guinea and Australian Aborigines, this is something like 1 to 3%. So a larger contribution of Denisovan DNA there. Now you can go to the next slide. So this was shifted a bit, but just to show you how we find this Neanderthal DNA in our genomes. And I find that a helpful way to think about it is to think about languages. So our genomes are languages, and Neanderthal DNA would be like borrow words. So, can you go one left? So here I'm showing you 4 Scandinavian languages: Icelandic, Swedish, Norwegian, and Danish. And you can see that they are quite similar, and especially the word weekend. This sentence is "have a nice weekend," and you see that Danish is the odd one out here. That's my language, and so Danish uses weekend, which is a borrowed word from English. So, and if you go to the next slide, so in DNA terms, that would mean that if we compare to Neanderthals DNA or Denisovan DNA, there will be some places in our genome where we look more similar to a Neanderthal than to other people. And those places we identify as Neanderthal DNA. It could also be Denisovan DNA in this example. Yeah, and then you would say that this little piece here is Neanderthal DNA. So now we can use this method that we talked about in the introduction to find look at a lot of genomes for modern humans and find all the places in the genome where there's Neanderthal or Denisovan DNA using that method I just talked about. And next slide. And so everyone will have a few percent of Neanderthal DNA and a Denisovan DNA, but those few percent will not be in the same places in a genome. It will be like different places. So if you had enough people, you can think of all these little Neanderthal fragments as puzzle pieces. You could actually piece together the entire genome of Neanderthals or Denisovans or any other type of human that we met with. And if you knew everything there was to know about how genes worked, you could infer exactly what they were like genetically, what they look like perhaps, and perhaps also some behavioral traits, just from looking at DNA from modern humans. So now let's just look at how much DNA there is in different groups in the world. So here I'm showing you just a bar plot of how much Neanderthal there is. So, and every little line is an individual, and I color them by where they're from in the world. So you can see that for Neanderthal, the most Neanderthal sequence is found in Oceania. Then there's a lot of Neanderthal found in East Asia and South Asia. And then there's less Neanderthal found in Europeans and people from the Middle East. But it's fairly homogeneous. But if you look at the Denisovan DNA, you see something quite different. You see huge differences in the genome. So you have a lot of Denisovan DNA in people from Oceania. Then you have the second most in people from South Asia, then East Asia, America, and again, very little in Europe and East Asia. And just to show you how much of this puzzle we can piece together, here I'm showing you how much of the puzzle is complete when you add more individuals. So the x-axis here is like if I just kept on sequencing individuals, and these are Icelanders, how much Neanderthal is the lower box, and how much Denisovan would I get? And you can see I'm sequencing 27,000 people, so quite a lot of people. But if you go to the next slide, you can see that if we do the same thing in India, which is a project I'm currently working on, with much less people, we can assemble much more of the Neanderthal and the Denisovan genome. There's something like only 10% of the data 10 times less data in India than in Iceland in this particular study. But there's much more Neanderthal and Denisovan diversity because there's much more genetic diversity in India compared to Iceland. So there is really a lot of DNA, and we can almost piece together entire genomes of Neanderthal and Denisovan DNA. But what effect does this have? What is the impact of these pieces of DNA? If you go to the next slide. So one very prominent example of a piece of DNA that has an impact is a piece of Denisovan DNA that's very common in people from Tibet. And I don't know if you have ever been in high altitudes or if you've just been hiking in the mountains, you realize you quickly become out of breath because the air is thinner up there. And that means that you get less oxygen. So sort of to deal with that, what your body does is that it produces more red blood cells. So you can absorb more oxygen. And this is a short-term solution, because the problem with that is that when you produce more red blood cells, your blood becomes thicker. And that puts a lot of strain on your heart. And if you do very intense activities, like giving birth, for instance, this can be very dangerous. But there is a piece of DNA that people from Tibet got from the Denisovans that help with this. Like it makes you produce more blood cells, but it doesn't make your blood as thick. So you sort of get out of this issue. And this is interesting because it suggests that, this Denisovan population that our ancestors met might have been living in high altitudes because they were supposed to be adapted to that. They had a version of a gene that helped them there, and then passed it on to us. And if you can go to the next slide. So another very prominent example of a positive impact that Neanderthal and Denisovan DNA has had on modern humans is they have boosted our immune system. And so here I'm showing you a different map of the prevalence of different types of infectious diseases all over the world. And this is what it looks like today. You could imagine that it looked very different 50,000 years ago when humans were first leaving Africa. And so you could imagine that when you're coming into a new environment, the Neanderthals and the Denisovans who were already living there were adapted to the local pathogens. So getting some of their immune genes might have been really beneficial. And that's why we see that a lot of our immune genes today are coming from Neanderthals and Denisovans. There's also a more sad example of integration. It's probably more of this did something else back then. So let me explain what I mean. So you probably all remember a few years ago when the pandemic was at its highest, and what researchers found there is that, and this is before vaccinations, that there is a piece of Neanderthal DNA that if you have this piece of DNA, your risk of being hospitalized for COVID is 50% higher than if you didn't have that gene. And I'm sort of showing you a world map here with how common this gene is. So the red little dots in the pie diagrams are how common it is. So it can go all the way to 100%. So it's very common in South Asia and Papua New Guinea, intermediate in Europe, and very rare in Africa. So COVID, of course, wasn't around 50,000 years ago. So this is probably a gene that had something to do with some other infectious disease. But nevertheless, in today's context, it wasn't a good thing. So there are some good things and some bad things that have today, some good and the bad effects of our health that have come out of Neanderthal and Denisovan integration. And another part of what we are currently working on is we are trying to figure out, okay, so we got a lot of Neanderthal DNA and Denisovan DNA. We can piece together this puzzle of, like, we can intentionally build the entire genome from Neanderthals and Denisovans. But are there some places in our genomes where there's never any Neanderthal DNA or never any Denisovan DNA? In other words, are there part of our genomes that are uniquely human, something that makes us different from them? And if you go to the next slide. So what I'm showing you here is our entire genome, all the chromosomes lined up. And all the orange places is sort of where the puzzle pieces overlap. So all the orange is all the parts of the Neanderthal and Denisovan genome that we could assemble by studying all these modern human genomes. But it turns out that there are actually a few places where we never see any Neanderthal DNA, we never see any Denisovan DNA. And those places are highlighted here in the blue bars. So there's one on chromosome 3, one on chromosome 7, chromosome 8, 18, and also the X chromosome has very little Neanderthal and Denisovan DNA, which is very interesting. Like, we don't know why this is. We don't know what genes are in these regions and what effect these regions have. So why a Neanderthal Denisovan DNA doesn't work in the human context. But that's an area of ongoing research. And yes, so that was that went a little bit quicker than I thought it would. But yeah, so I want to thank you all for listening and hearing about this little piece of the puzzle that Neanderthals and Denisovans gave to modern humans. Thank you.
Wonderful, thank you. We already have a bunch of questions coming in, so that was an excellent talk, and I think we will dive right into Q&A. So I think the first question, maybe like a sort of bigger question about sharing DNA. So David asks, can you clarify what is being measured when we say that we have 2% Neanderthal DNA, but 98% chimp DNA, and what does in common mean in these scenarios?
Yes. Yeah, so those things are measuring different things. Yeah. So let's do the human Neanderthal first. So you remember I said you in that example with the language is like this little piece of DNA is Neanderthal. So if you just, let's say the genome is just 10 base pairs long and Neanderthal is 1 base pair of that would be 10%. Like, so it's just what proportion of our genome is more like Neanderthal than other humans. For the chimpanzee and human chimpanzee, that's another thing they're measuring. They are measuring in genes how many positions in genes are similar. So it's a different measure. You could also have a measure of, for instance, like someone can be like 30% French, like 20% English, a few percent. Like, there's a lot of different things you can measure. That's also measuring long stretches of DNA.
All right, thank you. Here's another question about Neanderthal DNA. So Lauren asks, is it likely that the blue eyes, I think that our blue eyes, is due to Neanderthal DNA in us?
Not that I'm aware of. So the eye color is also one of those things that there are many different genes affecting eye color. I think it's something like 12 genes or something. And the combination of what version of each gene you have determines your eye color. So this blue eye color thing is something that when we look at ancient DNA from humans, so much more recent, is thought to have originated in the central steppe in Asia. So blue eye colors is very common in Russia, it's very common in Estonia, it's very common in Scandinavia and Northern Europe. But that's a more recent thing. It doesn't have anything to do with Neanderthals. It comes from a particular group of humans that had where more people have blue eyes that then spread into Europe.
Okay, thank you. Here's a question from Niels who asks, is there relatively little Neanderthal DNA in African populations, or has that just not been studied as much? So
there is, it actually wasn't studied until recently, and there is less Neanderthal DNA. There is some, but there is less. So there is, because humans and Neanderthals share a common ancestor, just because of that fact, we're gonna share some DNA. In addition to that, all non-African has much more. So there's some, but it gets even more complicated because we have this idea of like this out of Africa event, like some groups of humans leave Africa, some choose to stay, but it's more complicated because after meeting the Neanderthals, a lot of humans move back into Africa. So there is this back and forth gene flow, and they actually bring Neanderthal DNA with them. back into some populations in Africa. So you would say like all humans have some Neanderthal DNA, it's just in different proportions in different populations.
Okay. So this question from Urate follows on nicely from that, I think. So she asks, what about DNA from Homo sapiens that left Africa before 50,000 years ago? Can you distinguish that in modern humans? And then a second part of this question is, people speak of a ghost species seen in modern humans. What is this?
Yes, so let's do the first, that's a very good question. Let's do the first part of it first.
Okay.
So there are a lot of fossil evidence and also other evidence of humans outside of Africa before 50,000 years ago. So that's very likely. Like, in fact, like these populations might even have given some DNA to Neanderthals, it seems. But these populations, if we just say 100,000 versus 50,000, Neanderthals and humans are still much more different than that. They have a common ancestor, something like 500,000 years ago. So it still is not going to look very Neanderthal-like. It's going to look more different than between other non-African populations, but you can still distinguish that from Neanderthal. And the other question is an interesting one. So that was about these ghost populations.
Yeah.
So a ghost population is it's something that, for instance, if we didn't have Neanderthal or Denisovan genomes, we would say there are some pieces in our DNA that look very different, but we don't know where they're coming from. They just look different. And that's because we don't have fossil evidence. Then it's called a ghost population. So at some point, Neanderthal and Denisovan DNA were considered ghost populations that contribute DNA to modern humans. So there's a lot we still don't know. So we've managed to sequence the genome of 2 types of humans, and they both seem to have contributed DNA to us. What's probably very likely is that there were many other human groups around, and they also contributed DNA to us. We just haven't found that DNA yet. For instance, there's some people who would argue that there might also have been, other human groups living in Africa, not Homo sapiens, who contributed DNA to people who now live in Africa. The trouble here is that we actually, when we go further back than 50,000 years ago, we don't know a lot about human history. We don't know where exactly in Africa and like how many Homo sapiens populations there even were in Africa and how they were mixing and everything. So that question is a bit more hard to answer. But that is something that's being currently studied. And so hopefully we will get less and less ghost populations and more DNA from the actual populations.
So 2 similar questions that I think follow on nicely on that. Diane asks, with improvements in technology, is it possible that we will be able to extract and analyze DNA from other archaic humans? And Matt asked something similar. What's the likelihood that we'll be able to obtain samples of DNA from these other hominin species?
So the reason why, so I don't know if you remember the map that I showed you where the fossils are from, that we got DNA from. Most of them are from Siberia in Russia because it's cold. And because that makes DNA, the DNA breaks down when an organism dies, but it breaks down slower in colder climates. So the problem is that a lot of these other types of humans, like the hobbit, like Homo naledi, like Homo erectus, all lived in warmer environments, and some of them even hot and humid environments. And that's really tricky for DNA because it breaks down so much that in the end, it's very hard to analyze. But advances are being made. People are able to get better and better at getting DNA out of segments, and you're going further and further back. I think you're back to 10,000 years in Africa where you can get ancient DNA and slowly moving backwards. In addition to this, there's also the whole field of, so we're looking at DNA here. You could also look at proteins. So there's a whole field of proteomics where proteins also preserve, and they preserve better than DNA. So this might be our chance at getting some genetic information from these more places. So never say never. I think the future will hold some exciting new finds.
I like that optimism. So here's a question from Chris. Is there a settled view on how many Neanderthals and Denisovans and Homo sapiens there were at 100,000 years ago or 50,000 years ago?
Yes. So so one of the amazing things about DNA is, so your DNA you will get one chromosome from your dad, one from your mom. Your dad's chromosome will, like, some of it will be from his mother and his father, and so it goes back. So every single person in them is a living record of all their ancestors. And using that, you can actually look at, well, how different is it? And you can sort of get an idea of how big was the ancestral population, how many humans, for instance, were living. And what we see is that there were more humans when they left Africa. And it looks like that Neanderthals and Denisovans were living in smaller populations. So that's, some people speculate that might be one of the reasons that they're now extinct. It's like they were much smaller, more isolated populations than modern humans. But a number is hard to get.
Yeah, so it sounds like maybe relative population size, but getting a number estimate is hard.
Yes.
So we have a question, speaking of extinction, a few people have asked this. So Anne asks, is there any genetic information as to why Neanderthals and Denisovans became extinct and Homo sapiens prevailed?
Yes, so maybe here it's important to say that the answer to this question has changed a lot. And, you know, sometimes scientists' views sort of like is more a reflection of their own held beliefs. So if we go back to like 100 years ago, when the Neanderthals were first discovered, it was very common to think that, you know, because they're extinct, Neanderthals must have been very stupid or not very good at adapting to anything. And us humans, like being very superior, could easily like have outmaneuvered them. And that has been like, that has been holding, that idea of Neanderthals have been there for a while, like slowly starting to shift now. So there's no reason to say that they have some genes that make them less than us. There is, so there's no clear evidence of like, they had this gene, therefore they didn't survive, and we did. There is some evidence of, because they were living in smaller groups, they had slightly more deleterious mutations, but not something that would make them go extinct.
Okay. Yeah. Thank you. I think this question of why did any species go extinct in the past is always a kind of a bit of a mystery.
Yes. And it's also, most species will go have gone extinct. Like it's when you look at humans, like back 50,000 years ago, and you look at Neanderthal communities, you know, it's not like they're not so different. Neanderthals also care for their elderly, like they also seem to produce art. They also they're very good at like, very adapted to the environment. Like they can use very much different food sources. They are very good at making tools and sophisticated tools and knowing what stones to use and everything. So, you know, like there's no good reason to think that we were more than them.
Exactly. So here's a question from Muriel. Can you tell whether the Neanderthal or Denisovan genes came from the mother or the father?
Short answer is no. People have been looking into this question. So at some point, it was believed that it was only Neanderthal males who contributed DNA to humans. And the reason for that is that there's some part of the DNA that's contributed from mothers. It's called the mitochondria DNA. And there was no Neanderthal mitochondria DNA in modern human population. So people thought that means that mothers could not have contributed. So therefore, it could only have been Neanderthal males who contributed. What it turned out was that there's also a piece of DNA that's contributed only from males. And that's the Y chromosome. That's inherited from father to son. And there's also no Neanderthal Y chromosome in modern human populations. So that would mean that they didn't contribute either. So this argument is not very good. So you cannot say that it was only mothers or it was only fathers. But it definitely did happen. It's just neither the mitochondria or Y chromosome survives today.
Okay, thank you. So here's a question about methods. So Stephen
Mm.
asks, what's better for extracting DNA, bone or teeth or something else?
Depends. So generally speaking, some of the best places for DNA extraction, and this is gonna be a general thing.
Mhm.
What people have observed is that teeth are really good at it, and some bones from the body. So the petrous bone from the ear, one little bone there, is very good at preserving DNA.
Mhm.
That being said, like I just showed you Neanderthal Denisovan DNA from the little pinky bone and Neanderthal DNA from the toe bone.
Mhm.
And so it's hard to say. It depends very much where the bone was found. And even within the bone, when I was in Germany, like in Leipzig, where they extract a lot of the DNA,
Mhm.
they had examples where so what you do is like you take the bone and you drill into it and you get some bone part out and you get the DNA out of that. But it turned out if you drill one place, there's no DNA. If you drill another place in the same bone, there is a lot of DNA. So it's also not preserved equally in the bones. And exactly why it's preserved in some places and not others is something that we don't know yet.
Okay, interesting. I have a good follow-on question from the one you just answered, from Urate, who said, could you describe in layman terms, what's the process by which you get this information? So after the physical materials extracted, then there's a complex lab work. What are the steps in that work? And then there's statistical processing. How much standardization is there in these statistics?
Yeah, so I'm a bioinformatician, I'm a computer scientist. So when I enter this whole picture, the lab work is already done. So
Fair.
my description
Okay.
then is going to be short. So,
okay.
so I can tell you that you get the bone, of course, like you drill a hole, you get the powder out, and this powder is going to be mostly bacterial DNA. It's going to be contaminant DNA. So what I learned when I started was that I don't think they do it anymore, but back in the day, archaeologists would use to lick bones to say if it's bone or a rock, which would heavily contaminate the DNA with human DNA, the Neanderthal DNA. So you have all this DNA, most of it bacterial, most of it contamination. So then begins the whole process of finding pieces of Neanderthal DNA. And that's actually very hard. So one of the things you could say is that you can use some things. So Neanderthal DNA, it's old, so it's been broken down. That's one thing you could look at. And this breaking down of DNA also leaves some particular types of DNA damages on it that we don't see in modern DNA. So taking those things, you can sort of say, oh, I think this part of DNA is Neanderthal and not modern.
Okay.
So that's that process. Then when you've done that, you have all these little pieces of DNA, you gotta fit them to the genome. And the genome, as I told you, was a book of 3 billion base pair links. So imagine finding a little sentence of 10 letters and figuring out where in the book that fits. That's also not an easy problem.
You can't just copy paste that, right?
No. You gotta optimize and figure out exactly where it is. And once you do that, you get some. You can filter that and say, I only want to keep those where I'm really sure this is actually where it maps. And then where the little piece of DNA fits in the book. And then you can compare to, if you use the book metaphor again, human books. And then what you see is that then you see this, that Neanderthals are more different than humans are from each other. Like a Neanderthal and human is more different. But yeah, this is a process that's been developed over the past 20 years and a very complicated one. And one where assumptions are made. But the assumptions that are made, people try to take them into account. So it's not going to affect any of the conclusions that I talked about today, for instance.
Okay. No, and it's nice when people are explicit about their assumptions so we can evaluate that too.
Yes, there's a lot of things. Yeah.
Exactly. So here's a question from Steve that you partially answered, but just in case there's anything else in there, he asks, what does the genetic diversity in Neanderthals and Denisovans tell us about population size, which you talked about a little bit already, likely inbreeding and susceptibility to rare diseases?
Yes, so that could for sure be something that contributed to that demise. So just to give you an example, so a few years ago, I worked on a little group of Neanderthals from Chaghiskaya cave in Siberia too. And we found, I think it was 13 Neanderthals from the same cave.
Mm-hmm.
And then when we looked at their DNA, we saw that they were very similar, all the pieces of DNA. So similar that it looked like a very small population. And there was a lot of inbreeding too. There was a lot of that happening, more than you see in any, well, so the Neanderthals have more than Denisovans, and then even modern humans, hunter-gatherers from 30,000, 40,000 years ago, they also have much less inbreeding. So they were living in small groups, but, and you know, like, that means that you don't have so much genetic diversity. So if something changes, you are not as ready to deal with that. Like if new pathogens come in, for instance, like you don't have a lot of diversity in your immune genes. One important caveat there to mention is that this little population of Neanderthals that we looked at is from the easternmost part of the Neanderthal range. They were probably like much further away from Europe. So this fact that they were living in very small groups could just have been a specific thing to them, because they were living sort of on the fringe of Neanderthal territory. But for sure, this could have had an impact. And you could imagine maybe modern humans brought with them some disease that they were adapted to, that the Neanderthals were not adapted to.
So speaking of adaptation, Cindy asks, it's very interesting to hear about the impact of, she writes, and I think it's maybe Neanderthals on COVID and living at high altitudes. What other advantages or disadvantages does the Neanderthal genome provide humans?
Yes, so there's actually a lot, like there's been done a lot of studies figuring out what genes do and what effect they have. Is also a very tricky thing, because many things are caused by the interplay of a lot of genes together. For instance, one thing that has also been shown is that Neanderthals contribute to height. They make you a little shorter. So, and which makes sense, like Neanderthals, the fossils that we found were shorter than the modern humans, shorter and more sturdy built. But this is a very small effect because there are so many genes contributing to height. So it's just one gene that makes you a little less shorter, but there's so many other genes that could have an effect. So that's a relatively small effect. Another example is that there is a Neanderthal part of the DNA that reduces your risk of contracting prostate cancer if you are a man. So, I mean, that's a nice thing. People have also actually with the COVID thing. So there is this piece of DNA that increases your risk of hospitalization. Independent of this, somewhere else in the genome, there's another little piece of DNA, the Neanderthal DNA, that actually makes you a bit more protected against COVID. So it's just this one that I mentioned, it has the strongest effect, but the other one is also true. So it's not even that Neanderthals, the DNA that we got from them, makes us more susceptible to COVID. Like it makes us more susceptible, but also a little less at the same time. Things like, you know, people have been trying to look at what if we see Neanderthal versions of genes in humans and we know what humans look like, could we maybe infer what Neanderthals look like? This is a tricky thing. So people are trying to infer that Neanderthals have red hair, for instance, and the skin color was maybe lighter. This is a tricky thing, and I would take this with a grain of salt. It's very hard genetically to predict these things, and we can't even do it in modern human populations sometimes. So what Neanderthals look like is still something that we still don't really know.
That's a good note of caution. Thank you.
Yeah, we have to wait until maybe we find a Neanderthal in the permafrost somewhere.
Oh, that would be amazing.
To see what they actually look like. Yeah.
So a couple of more methods questions. So Stephen asks, how many different specimens of Denisovans or Neanderthals are available to study?
So we could maybe say that sometimes you get a lot of DNA and you can get a lot of genetic information, and you call this like a high quality ancient DNA sample. Of those, there are 3 Neanderthals and there's 1 Denisovan. Only 1.
Oh, wow.
So far.
Okay.
There is something like, if you can also count all those where the DNA wasn't preserved very well. So the quality of DNA you can get out is much lower. And if you count that, it's something like 30 Neanderthals or something like that.
All right.
Without spoiling too much, I can say that there will be more Denisovan genomes and Neanderthal genomes in the future.
That's exciting.
I'm sure of that.
I think this may be a question that might have a similar answer. So Diane asked, you mentioned that many thousands of Icelanders' DNA have been studied. So she asked how many individual Neanderthals and Denisovans' DNA are being used as a benchmark for their species. So I think you sort of just answered that one.
Yeah.
So
3 Neanderthals, 1 Denisovan versus, like Iceland, there's a lot of people who have their genome sequence, but in the whole world, it's maybe 1 million people who have their DNA sequence that we can study.
Okay.
So yeah, very uneven sample sizes.
Yeah, exactly. So here's an interesting question from Bill. We hear a lot about the effects of Neanderthal DNA on modern humans. Is there any data about the effects of our DNA or Denisovan DNA on Neanderthals?
Yes, so that one is a bit harder because with modern humans, you know, like you have a person and you can measure things about that person. You can say, like for instance, do you have higher blood pressure, or like how tall are you, or what do you look like, or something like that. Those things are very hard to figure out about Neanderthals and Denisovans. So because we only have very few pieces of bone left, so if it had some impact, it would have to be something we could observe in those bones. Yeah. You know, like some disease that does something to the bone, or something that makes you way shorter or taller, for instance. Interestingly enough, there is evidence of, as I think I talked about earlier, that there's been earlier migrations out of Africa by modern humans. And there is good evidence that they actually contributed a bit of DNA to Neanderthals too. Interestingly enough, there's these regions in the genome where there's never any Neanderthal DNA in modern humans. We can call them genetic deserts for now. Those are also in Neanderthals. They never have any human DNA there. Maybe suggesting that this was a region where if you get some DNA from another context, it's not beneficial. There's something in these regions that are very specific to each of the groups. What that would be, we don't know yet.
So I think this question follows on that maybe. So Urate asks, what's the function of those spots on the 4 chromosomes on which there's no Neanderthal or Denisovan DNA?
I mean, if I knew, I would tell you. Yeah. That's a very good question. So we don't know yet. So, you know, you can sort of look at those regions like, are there, for instance, you can ask, like, are there more genes in these regions than other places in the genome? And there seems to be more genes there. Okay, so is it just the number of genes? Does that do something? Or is it maybe more, what do these genes do? So people have been trying to look at that. So a lot of the genes there are, so genes can have different roles in different cells. You know, your cell types are different. You can have skin cells, brain cells, heart cells. So it seems that some of these genes have a big effect in the testes. So this has something to do with like reproduction. So is that why it has another effect, and so that maybe makes you less able to have kids? That would be a huge thing to be selected against. Yeah. But to tell you the truth, we're still working on figuring out what these regions do.
So I think this question maybe touches on this a little bit from Kay, who asks, has there been any new or decisive evidence that sperm in Neanderthals became less viable?
Not that I'm aware of. So it's hard. So, I mean, you can get closer and closer to answering that with all these new methods. So you can essentially like construct a Neanderthal genome, and maybe you could imagine some experiment where you can make some sperm cells that have Neanderthal DNA and human DNA and seeing there's any difference in how well they survive in a petri dish or something like that. They're still a bit out, I think.
So here's a pretty specific question from Michael, who asks, considering the research to date, do you think that the archaic ingression found mostly on autosomal portions and rarely on the X chromosome is more a consequence of hybrid incompatibility or sex-biased introgression events?
That's a really good question. So some people have actually been working on that study, studying that, and so there's very lot of interesting things going on the sex chromosome. So that study with, is it because males are contributing more or less than females?
Mm-hmm.
That one is hard to say, because it's hard to entangle what happened then versus what happened after the next 50,000 years in human history. It seems like, I forget what the conclusion is. I think it is that it's slightly more male driven, but not like only males, only females, something like that. The hybrid incompatibilities is really a really interesting hypothesis, because you see that in other groups of animals, where if you have, for instance, if a lion and a tiger will have an offspring, and it's male, that will not be able to produce more offspring. Is that a thing that's happening in humans and Neanderthals? I mean, probably not, because we managed to have kids, like the DNA lives on in us, so not to the same extent at least, but some of the forces at play there could also be at play here, but in a smaller scale.
Here's an interesting question, thinking about things we can see from ancient genes, maybe. So Stephen asks, have we found specimens of Neanderthals or Denisovans that had, quote, modern diseases like cancer or TB or syphilis?
Not that I am aware of yet. I'm just trying to think, like, so there's definitely some Neanderthal bones where you look at them and it looks like they did have something in the bones, but if it's a modern human thing and how common this is, I couldn't tell you. So to my knowledge, not yet.
Okay. Here's a question from Najas. Don't know if I'm pronouncing your name correctly. Is it the case that different populations of modern humans carry different parts of Neanderthal or Denisovan DNA?
The answer would be yes. So for instance, there's a lot of Denisovan DNA in Australian Aborigines and in Papua New Guinea that is not found in other places of the world. So these Denisovan pieces of DNA would be unique to them, or not found in other world. Like we haven't studied everyone's genome. They might be around other places, just very, very rare. And there's also the same thing with if you look at Europeans versus East Asians, they also have some Denisovan DNA. For instance, the high altitude one that is not found in Europe. So there's also a unique piece that's unique to them. And then what's very interesting is the study I'm working on now, where we are looking at the genomes of a lot of people from India, is that there seems to be a lot of Neanderthal and Denisovan that we haven't seen anywhere else in the world yet. And this sort of like gets at the question, so where did we meet Neanderthals and Denisovans? Was it maybe not in the Middle East? Was it maybe somewhere else, more in East Asian-ish, where there's more Neanderthals and Denisovans living? And yeah, so we're not quite sure about that either yet.
Interesting. So here's two questions about like specific functions of Neanderthal or Denisovan genes. So Selquist asked, is life expectancy different depending on the amount of Neanderthal or Denisovan genes? So why don't we start with that one and then we'll go on to the next one.
No, there isn't a difference. And this is also a difficult question to get at, because life expectancy is, so there's always a combination of your genes and the environment. And this life expectancy one particular is going to be very affected by your environment too. And if you study people from, East Asia, or let's take Oceania as an example, Papua New Guinea versus Europe, the environments are not the same. So it's very hard to tease out the effect of just the Neanderthal Denisovan DNA. But there's no evidence for that. So don't worry if you have if you take a 23andMe and you have a lot of Neanderthal, I think you will be fine.
That's reassuring.
Yeah.
And so another question about specific functions. So Cindy asks, is there a relationship between the risk of having osteoporosis and the percent of Neanderthal DNA?
The answer to that is I don't know.
Mhm.
I couldn't say.
Okay.
I'm not aware of it, but that doesn't mean that it's not true. Like I'm not aware of everything that's happening, unfortunately.
Sure.
But a good
That's the
question.
nature of
Yeah.
science. There's a lot of
Yeah.
amazing discoveries happening. So here's an interesting question, another one from Urate. So Svante Pabo speculated that DNA differences between Neanderthals and modern humans indicated maybe some differences in brain function. Do you have any comments about that work?
Yeah, so actually, like when it comes to it, Neanderthals have bigger brains than we do, than modern humans do.
Mhm.
And it is possible that something different was happening. So I talked about these deserts with no Neanderthal DNA. So originally, there was a gene in one of those deserts that looked very promising, like an explanation for why maybe that would be not good to have Neanderthal DNA there. And this gene is a gene involved in language. So there is a family in the UK who just by chance have a mutation in that gene. So it doesn't work as well. And what happens to them is that cognitively they are normal functioning, but they have a very hard time constructing sentences. So people are speculating, is that maybe a human thing? Is it because like Neanderthals, they had a hard time having the abstraction needed to make sentences and like plan that out. It turned out later that if you sequence the Neanderthal version of that gene and the human one, they're identical, like they're not different. So it's more complicated than that, but yeah, I know that there's also a lot of work on, so it's hard to say, there's also a lot of work on, you're trying to introduce Neanderthal variants into human brain cells, like not living people, just brain organoids, and they seem to function somewhat differently. The problem is, of course, always you cannot reconstruct the entire brain and seeing how that all works. So it's a hard thing to answer, but there might have been differences, but it's just, what did these differences mean? Did they have any meaningful impact?
Yeah.
It's
So we still have a bunch of questions, but I'm going to ask one last one before we wrap up. And this is a question for Matt. What new developments or possibilities in this field are you looking forward to? What's next?
Oh, what's next? So there was at least 7 different types of humans alive. And now we have DNA from, 3 of them. Humans, Neanderthals, and Denisovans. But how many more are there? We were just, I wasn't involved. Someone, an archaeologist was working in a cave, found a little bone. It's an entire new group of humans. How many more are there? Like, how complicated is our family tree actually? And, like, what were these populations like? And I think we're gonna get much closer in understanding this in the coming years. So I'm looking forward to that.
That's very exciting. Mysteries, hopefully we'll get closer to solving. So thank you. So I'm going to wrap up today's virtual program. Please join me in thanking Laurits for sharing his work with us. I'd also like to give special thanks to those who made this program possible, to our behind the scenes team who helped sort through your questions, including Dr. Ryan McRae from the Smithsonian's Human Origins Program, to our donors, volunteers, and viewers like you. And finally, to all our partners who help us reach, educate, and empower millions of people around the world today and every day. We thank you. So next month will be our last Hot Topic program for 2023 on December 14th at 11:30 a.m. A colorful path, what the ancient pigment ochre tells us about the modern human mind, with archaeologist Tammy Hodgkiss from the University of the Witwatersrand in South Africa. We've put a link in the Q&A where you can find information about our upcoming programs and how to sign up for the museum's weekly e-newsletter. That's the best way to stay informed on our upcoming programs and to learn more about the museum's research and exhibitions. After this webinar ends in a moment, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond. We're very curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. Again, Thank you to Laurits, to our participants, and to you, the audience. And we'll see you next month.